Hello everyone, I am Andrea Luppi from the University of Cambridge, and I will be talking to you about some work that we did on the connectome harmonic signatures of consciousness. So let me introduce you first of all to the harmonics themselves. So I hope you noticed that these beautiful patterns of sound moving over a vibrating metal plate became increasingly complex as the frequency of the corresponding sound was increasing. And you can see here on the screen the same. And crucially, this is not just something that happens on metal plates, because this is also the basis for how we make music. As you can see here, increasingly complex patterns of vibration take place when frequencies increase, for instance, over the body of a guitar. But this is not just something that happens in artificial systems, because the very same phenomenon can also explain the formation of the patterns of pigmentation over the coats of various animals, as Murray and colleagues demonstrated beautifully. And importantly, all of these phenomena can be captured by one and the same equation. And this is shown here. And that equation is saying that the diffusion over a medium, for instance, diffusion of sand or diffusion of pigmentation patterns over the coats of animals, can be captured in terms of spatial patterns, which we refer to as harmonics, and their corresponding periodicity with which they manifest. And so we were wondering, can the same phenomenon also capture information flow over the medium of the human connectome, that is the network connecting brain regions? And the answer is yes. And you can see here this beautiful set of harmonics derived from the human connectome, which Atasoy and her colleagues obtained in 2016. And we refer to them as connectome harmonics. And as you can see here, there is a striking resemblance between the connectome harmonics and the patterns of sound vibrating over a metal plate, which are obtained completely differently and yet observe the same equation. Crucially, you can use these connectome harmonics as a new language to represent brain function. And this is similar to the Fourier transform applied rather than in terms of sine waves, in terms of the human connectome, that is the graph connecting human brain regions. And a different way of thinking about this is in terms of finding the ingredients of your functional brain activity over time, much in the same way as you would find ingredients of your smoothie. And using this technique, which is called connectome harmonic decomposition, you can decompose your functional brain activity into its corresponding harmonics. Specifically, you can talk about this in terms of energy, which is the normalized contribution of each harmonic to the brain activity data that you have. And using connectome harmonic decomposition, Atasoy and colleagues were able to demonstrate that both LSD and another serotonergic psychedelic known as psilocybin, which is the psychedelic compound found in magic mushrooms, have a particular signature in terms of connectome harmonics. That is, they show decreased energy over the low frequency harmonics, which are the ones that explain most of the variance due to large scale patterns in the human connectome. And instead, LSD and psilocybin show both increased energy over the high frequency harmonics, which are the ones corresponding to more fine grained and more complex patterns. And in the work that we did here, we showed that the same phenomenon also applies to a different kind of psychedelic, that is ketamine, which operates on NMDA receptors rather than serotonergic receptors. And here we had given our participants subanesthetic doses of psychedelics, which induce phenomena of a psychedelic nature. So we show that this signature can generalize not just to serotonergic psychedelics, but to the psychedelic state in general. And interestingly, the opposite signature is instead obtained when participants are anesthetized with the GABAergic agent propofol, which is increased energy over the low frequencies and instead decreased energy over the high frequencies. That is to say, the psychedelic state and the anesthetized state, so increased, if you will, an enhanced state of consciousness and a decreased state of consciousness in terms of connectome harmonics, are mirror reversed images of one another. Crucially, this kind of analysis is generalizable to any state of consciousness that you care about. And it doesn't even have to be a state of consciousness per se. Rather, any kind of brain imaging data that you have can be used to obtain a signature. And you do this by first decomposing the activity that you have, in this case from functional MRI, in terms of the corresponding harmonics. And you find what patterns of energy your data are decomposed into, and from those, you can obtain a particular state-specific signature. Now, in this particular case, for the data shown here, we see that the signature of this state corresponds to increased energy over the low-frequency harmonics and decreased energy over the high-frequency harmonics, which allows us to place it on a two-dimensional scale of high-frequency and low-frequency harmonic energy, and we see that it is right next to anesthesia, which is not surprising because the state that we were actually describing here is the comparison of patients with brain damage who are in a disorders of consciousness or those who do in fact have covert cognition, as demonstrated by their ability to play tennis. Imagine playing tennis in the scan. So conscious versus unconscious patients have the same signature as conscious versus anesthetized individuals who are otherwise healthy. And we can see that this pattern generalizes even further because we can take the pattern of brain activity that discriminates between patients with disorders of consciousness and patients who are actually able to play tennis in the scanner. Imagine playing tennis in the scanner. And this very same pattern can predict the amount of propofol that is necessary to bring someone into anesthesia or to bring them out of anesthesia and into recovery. So we can show all the way from disorders of consciousness to the amount of propofol in the blood, to a biological marker of the presence or absence of consciousness using this connectome harmonic decomposition. And so effectively what we can do is map a landscape of states of consciousness in terms of the energy of connectome harmonics over high frequency and over low frequency. And we can see here mapped some of the states that we have explored so far, so psychedelics, anesthesia, and disorders of consciousness. So to summarize, what we think we have found is a way to map the landscape of states of consciousness using the connectome harmonics as our compass. And if you want to find out more, you can come to see my virtual poster, which is number 2195 at virtual OHBM. So I will leave you with a quote from Nikola Tesla, who said, "If you want to find the secrets of the universe, think in terms of energy, frequency, and vibration." And I don't know about the universe, but I think that energy, frequency, and vibration might very well tell us something about consciousness. And of course, I would like to thank the organizers for inviting me, my funder, Skate Cambridge, for allowing me to perform this work, and my very many collaborators, as well as my supervisor, Dr. Emmanuel Stamatakis, and my advisor, Professor David Menon. And I will be happy to take any questions. Thank you.
Hi, my name is Catherine Duclos. I'm a postdoctoral fellow at McGill University. And the title of my talk today is Reconfiguration of Network Hubs Under Anesthesia May Predict Recovery of Consciousness in Unresponsive Patients. So before we begin, what is consciousness? Well, we generally assess consciousness along two main axes, wakefulness and awareness. So in full consciousness, an individual is both able to awaken and to be aware of his or her environment and of his or herself. On the opposite end of the spectrum, we have coma, where an individual is unable to awaken and is unaware of himself or his environment. Along the spectrum, we also have what are called disorders of consciousness, which include unresponsive wakefulness syndrome, previously known as vegetative state, which is a state in which a patient can awaken but is deemed unaware of his or her environment and of himself. We also have minimally conscious state, where we show intermittent signs of minimal consciousness. And we have locked-in syndrome, where an individual has brain damage that makes them unable to move, speak, or respond to stimuli. So they are pretty much locked in their body despite preserved consciousness. When an individual suffers a brain injury, their level of consciousness is generally impacted. They can be in a coma for several weeks to several months. They can then transition to a vegetative state, minimally conscious state, and if they're lucky, they will eventually emerge from a disorder of consciousness and regain full optimal consciousness. However, we do see brain injured patients who remain in a disorder of consciousness for many months to many years, and then can even remain in this state for the rest of their lives. Despite major advances in our understanding of the neural mechanisms of consciousness, clinical assessments of these patients still result in the misdiagnosis of disorders of consciousness in up to 40% of unresponsive patients. That is, 40% of individuals who score as being in a minimally conscious state on the coma recovery scale, which is our gold standard research scale, are diagnosed as being in unresponsive wakefulness syndrome. So even though they have minimal consciousness, they are diagnosed as being completely unconscious. This can have significant effects on level of care, treatment decisions, and access to rehabilitation. And what we know for certain is that unresponsiveness is not equal to unconsciousness. So what we need is to develop objective and quantifiable markers of consciousness in unresponsive patients. Recently, consciousness science has seen the emergence of a novel strategy of consciousness detection, and it's based on the quantification of the complexity of the brain's response to a direct cortical perturbation. So these studies using the perturbational complexity index have shown that the complexity of a brain's response to a direct cortical perturbation, in this case, transcranial magnetic stimulation, is strongly associated to the level of consciousness, whether we're speaking of sleep, anesthesia, or disorders of consciousness. However, since TMS is not an accessible tool to a majority of clinical teams for bedside assessment, it's not a tool that's easily translational to clinical practice. However, general anesthesia is an alternative approach for perturbing brain networks and to quantify their complexity, and it offers an accessible within-subject paradigm for the detection of consciousness in unresponsive individuals. Network neuroscience has shown that a healthy brain adaptively reconfigures its functional network under anesthesia. We know for one that alpha power shifts from the posterior to the anterior part of the brain under propofol anesthesia, but this is mainly an effect of the propofol itself. We also know that the anesthetic-induced loss of consciousness has a similar effect on network hubs, which are the most highly connected nodes, in this case, electrode channels within a network. So what we see at the bottom of this, what we see in this bottom figure is that alpha hubs also anteriorize under anesthesia in healthy individuals and then return to a posterior location upon recovery of consciousness. We therefore propose to measure brain activity using high-density EEG and to use propofol anesthesia as a way to perturb the brain. Our hypothesis is that the capacity for the reconfiguration of brain network hubs under anesthesia would be indicative of the brain's ability to support consciousness. So to test this hypothesis, we recruited 12 unresponsive patients in a disorder of consciousness following a brain injury. Average age was 42.6 years old. 7 of these patients were in the acute phase, that is within 2 months following injury, and 5 were in the chronic phase, ranging between 9 months and 21 years post-injury. We used 128 channel EEG to record EEG brain activity before, during, and after propofol anesthesia, and we assessed level of consciousness using the coma recovery scale revised. Patients were therefore in their unresponsive resting state for the 5-minute baseline recording. We then started the anesthesia using a target-controlled propofol infusion, and once we reached a target effect site concentration of 2 micrograms per milliliter, we did a second 5-minute EEG recording. We then stopped the propofol administration, waited until propofol concentration reached minimal levels, and then we carried out our third EEG recording at rest. We then bandpass filtered the data between 0.1 and 50 hertz. We re-referenced our data to an average reference, and we removed non-scalp and noisy channels, and also noisy epochs with non-physiological artifacts. We then constructed our alpha brain network by bandpass filtering into the alpha band. Our connectivity measure was the weighted phase lag index, which determines the degree of phase locking between two signals. So we calculated the weighted phase lag index, thresholded our data at the top 5% to keep only the strongest 5% connections to construct our network. And then we calculated the degree of each node, that is the number of connections of each node, in order to assess the location of the high degree network hubs. For the baseline recording, so the topographic configuration of alpha network hubs varied greatly between subjects. So for coma, unresponsive wakefulness syndrome, and minimally conscious states, some patients had hubs that were dispersed and seemed somewhat randomly organized. However, we also had subjects whose hubs looked more spatially coherent, and they were located in the posterior regions of the brain. Importantly, there didn't seem to be an association between hubs and the current level of consciousness, as the healthier looking hubs were found in 1 coma patient and 3 unresponsive wakefulness patient, rather than in the minimally conscious state patients. So among the 8 patients who had more random hub organization at baseline, propofol anesthesia resulted in 2 different patterns. So on the left side, we see 4 patients who barely showed any reorganization of hubs under anesthesia. And on the right side, we see individuals who showed some hub reorganization under anesthesia, but they either started at or reorganized to a somewhat unusual topography, or they had no clear return to their baseline topography during the recovery period. Importantly, out of these 8 individuals, none of them recovered consciousness following the EEG recording within the 6 months that followed. Interestingly, the 4 patients that had a clear reorganization of alpha network hubs that resembled healthy individuals are shown here. So they showed spatially coherent posterior hubs at baseline. These hubs anteriorized during anesthesia and then returned to their spatially coherent posterior location during the recovery period. And of these 4 subjects, the first was presumed to be in locked-in syndrome, which is a conscious state but an inability to respond, and he was withdrawn from care. And then the 3 other patients actually recovered consciousness within the weeks that followed this EEG recording. So overall, this preliminary study suggests that the coherence and topography of hubs are not directly associated with the current level of consciousness, as we saw with the baseline data, but they seem to predict recovery of consciousness in unresponsive patients. So they unveil the potential for the recovery of consciousness. Additionally, our results suggest that anesthesia is a promising tool to uncover the potential for consciousness in unresponsive patients. Future studies should aim for a larger sample size and should aim to develop an index that quantifies the amount of network reorganization induced by anesthesia. Ultimately, using anesthesia to assess adaptive network reconfiguration may complement clinical measures to improve diagnosis and prognosis of DOC patients, aid in clinical decision-making and end-of-life decisions, and ultimately improve rehabilitation opportunities and enhance quality of life in this population. And with that, I'd like to thank you for your attention. I'd like to thank my supervisor, Stephanie Blaine-Moujaes at McGill University, and also thank all the funding agencies that have supported our work and that are supporting my postdoctoral fellowship. Thank you.
This project aims to explore ketamine's influence on global resting state functional connectivity. So why are we interested in the effects of ketamine, an uncompetitive NMDA receptor antagonist on the brain? We know that the acute administration of ketamine, so the most commonly used dose in human research is around 0.5 milligrams per kilo, produces transient psychotic symptoms in otherwise healthy adults, which are listed here. One of the reasons there has been such an increased interest in the effects of ketamine is the discovery that while acute ketamine administration can induce transient psychotic symptoms in otherwise healthy adults, it can also act as a rapid-acting antidepressant in patients with major depressive disorder. What do we know so far about how ketamine affects the brain? We know that ketamine causes robust changes at the level of cells and microcircuits. For example, animal studies have shown that ketamine can reverse the loss of synapses caused by stress. By restoring the dendritic spines, ketamine restored the loss of functional coupling between neurons. Ketamine also has robust neural effects on large-scale functional connectivity. Here, we will focus on studies that have used a data-driven method called global brain connectivity to explore ketamines effects on the brain. To calculate global brain connectivity, or GBC, you take the time course of a time point, correlate the point's time course with every other point's time course, repeat this for all points, and then transform the correlations to Fisher Z values. So you're essentially computing the mean connectivity strength for each time point. This shows how globally coupled each point is to the rest of the brain. This study by Dresden found that ketamine increased global brain connectivity during rest, both 5 minutes after a ketamine infusion, pictured on the left, and 45 minutes after ketamine infusion, pictured on the right, showing that this increase in global brain connectivity following ketamine is a stable effect. They also found a number of clusters where increased global brain connectivity was associated with positive PANS symptoms, and a single cluster where decreased global brain connectivity during ketamine was associated with greater increases in negative symptoms. We were not able to replicate these results, possibly due to the fact that they used a much larger Gaussian kernel to smooth their data than we did, or possibly for another reason, which I will talk about during the results section. So here's an overview of the study. We recruited 28 men and 12 women. During the scan, first of all, a saline infusion was administered, and then a resting state scan and a TAR scan were commenced. Then a ketamine infusion was administered, which was an initial bolus of 0.23 milligrams per kilo, followed by a maintenance infusion of 0.58 milligrams per kilo per hour. And following the ketamine infusion, the participants then underwent another resting state scan and a TAR scan. As we were interested in the psychosis-like effect of ketamine, we collected PANS, which is the Positive and Negative Syndrome Scale, a medical scale used for measuring the symptom severity of patients with schizophrenia. We collected this before and after the scan. We also collected cognitive data during the scan using a spatial working memory task. All data was pre-processed according to the methods provided by the Human Connectome Project using QNX. So onto the behavioral results. We found really robust behavioral results, and these were retrospectively assessed 118 minutes after ketamine administration, and we found that there was a significant increase in negative, positive, and general symptoms as assessed by the Positive and Negative Syndrome Scale. And there was also a trend towards a decrease in depression as measured by Beck's depression inventory. Cognition was assessed by a spatial working memory task in the scanner, and we saw that ketamine resulted in a significant impairment in cognition compared to placebo. A neural GBC map for the pre and post ketamine sessions for each subject was calculated. Group level effects were then examined, comparing ketamine and placebo. So first of all, these are the results at the voxel level with 91,281 voxels. So you can see in the top, the placebo mean map, the ketamine mean map, then the ketamine minus placebo t-test, Z-score map, and finally in the bottom right, thresholded t-test map, which was type 1 error protected. And in the bottom right thresholded map, you can see that ketamine resulted in hyperconnectivity in subcortical areas in the left medial occipital cortex and hyperconnectivity in the right temporal cortex, right insular cortex, and right anterior singular cortex. Here you can see the results after the neural GBC maps were first parcellated using functionally defined parcels. So in this case, it was 718 parcels. And as you can see in the bottom right map, which is the ketamine minus placebo t-test type 1 error protected map, not much survives after correction for multiple comparisons. And finally, we conducted the same analysis, but first parcellating for networks. So here we parcellated the data into 12 networks. And in the thresholded map on the bottom right, you can see that ketamine led to increased global brain connectivity in the default mode network, which is in yellow, and the primary visual network in orange. And here you can see the network parcellation labels on the left, and on the right is a bar graph showing the change in mean connectivity following ketamine in each network so that you can better interpret the unthresholded results. However, what became apparent to us as we analyze the data is that there is a mismatch between the behavioral and neural results. So there are these robust behavioral differences, but we do not see strong group level differences. And this was especially true at the voxel level, which previous studies had reported on. So given that we couldn't replicate what was in the literature, we began to wonder whether, if this analysis makes an incorrect assumption, so whether it makes the assumption that the mean shift is the right solution, when in fact a multivariate approach may be more appropriate. So what if ketamine acts on multiple axes? To test this, we conducted an additional analysis using a multivariate approach, in this case a principal component analysis. So we first subtracted the placebo parcellated neural GBC map from the ketamine parcellated neural GBC map to produce the delta GBC neural map for each participant. We then conducted a principal component analysis on the delta GBC neural maps and found, as we predicted, that there was not just one dominant principal component. So here we see there are 8 principal components which were significant, with the first 2 both explaining a high amount of the variance. So here you can see the neural GBC map for PC1, which explains 12.3% of the variance, and PC2, which explains 9.9% of the variance. We show that the PC1 neural delta GBC map produced by the multivariate analysis, so the principal component analysis, did not correlate with the t-test map from the univariate analysis. As you can see, there was a correlation of minus 0.19. The same was true of the PC2 neural delta GBC map. So again, we see there's a very low correlation of minus 0.17. This indicated that the univariate analysis that essentially compares the mean neural GBC maps before and after ketamine may not be capturing the full effects of ketamine on the brain. Next, we wanted to connect the behavioral results to the neural results. So we mapped the change in behavioral symptoms following ketamine to the change in neural GBC following ketamine. To explore the relationship between individual symptoms and neural variation in response to ketamine, we used the neurobehavioral relationships in dimensional geometric embedding, or MBridge, framework developed by Lisa, a postdoc at the Antichevich lab. First of all, we assessed whether there was collinearity between the 31 behavioral measures, PANS, which is well known to be collinear, and our cognitive measure. Given the collinearity, we then explored, which you can see in the correlation matrix, we then explored whether there was a lower dimensional representation that could capture most of this natural symptom variation. To do this, we again performed a principal component analysis using all of the behavioral variables, and after permutation testing, the first 2 principal components were found to be significant. Together, they account for about 41.4% of the total behavioral variance in our dataset. Here you can see the loading of the 2 principal components. So PC1 seems to capture the general impairment caused by ketamine, and PC2 seems to capture impaired cognition, impulsivity, and uncooperativeness. We then mapped the behavioral principal component scores to the parcellated neural delta GBC maps via mass univariate regression. Here you can see the resulting neural map for the neurobehavioral PC1, and here you can see the resulting neural map for the neurobehavioral PC2. We then ran a correlation to test whether participants who score highly on the neural PCs also score highly on the neurobehavioral PCs. The results in the correlation matrix here show weak support for this hypothesis. This indicates that including the difference in behavioral symptoms when we created these neurobehavioral PCs provides us with new information about the neural data. Finally, we started to explore whether the effects of ketamine vary similarly to psychosis. So to do this, we used the MBridge framework to examine whether the axes of neural and symptom variation in response to ketamine vary similarly to those defined in psychosis spectrum disorders. So MBridge allows you to project data into a already defined space, brain behavior space. So we first projected behavioral and neuroimaging data from the 40 subjects pre and post ketamine into a behavioral brain geometry defined in an independent discovery sample of 436 psychosis spectrum patients. The idea is that you can then see how each person projects onto the psychosis principal component pre and post ketamine. We hypothesized that people will not load strongly onto the psychosis principal component on a placebo on the left, but will load strongly onto the psychosis principal component following ketamine on the right. Here are the 40 subjects projected onto the psychosis principal component, and the bar chart here allows you to see more clearly that ketamine does not affect everyone in the same way. A third of people project more positively onto the psychosis principal component following ketamine, while 2/3 of people project more negatively onto the psychosis principal component following ketamine. So we're still progressing with this analysis, But as for now, the key takeaway points so far are that ketamine seems to act on multiple axes, so a multivariate approach may be a more appropriate way to quantify its effects in the brain, and also that it is critical to characterize the mapping between single subject level changes in symptoms and neural features. Thank you so much for listening, and thank you to everyone in the TeacherBitch Lab for collecting the data, and also a big thanks to Dr. Lisa, Chee, Dr. Allen and TeacherBitch, and Dr. Katrin Preller for all your help and guidance with this analysis. So if you have any questions, please let me know.
So thank you for coming to my talk. The title is Slow Wave Sleep is an Altered, Not a Reduced State of Consciousness, Resting State Network Functional Connectivity in Sleep. I have no conflicts of interest to disclose. So one of the larger mysteries of sleep is why we need to be disconnected from reality. Instead of processing information from the outside world, our brains are largely sealed off in their own world for what amounts to 1/3 of our lives. So just what on earth is the brain doing with such an enormous amount of time? And why does it need to cut itself off from reality to do whatever it's doing? In order to get at these answers, we need a way to track brain activity during sleep. Until now, EEG has served us well in this regard, telling us that there are two broadly distinct patterns of activity. Low amplitude, high frequency oscillations in REM, which look suspiciously similar to wake, and large amplitude, slow frequency oscillations in non-REM, which increase from non-REM 1 to non-REM 2 to slow wave sleep. Now the problem, of course, is relating this activity to function. Because the brain is largely cut off from the outside world, it's challenging to design studies that can help us relate activity to function. In waking studies, we make the assumption that the subject is doing what we've asked them to do. In sleep, that key assumption falls apart. But what if there were a way to get around this problem by instead looking at the activity of brain networks that have already been tied to function during wake? As it happens, there's an amazing phenomenon particular to fMRI called resting state networks. Resting state networks have a strong spatial similarity to regions of the brain that have already been associated with cognitive functions. For example, this auditory network is so called because it maps well onto areas of the brain known to be associated with auditory processing. The following networks are all related to specific kinds of sensory processing. Other networks map well onto brain regions associated with the processing of higher order information. For example, the default mode network is active during daydreaming and internally directed thoughts. The remaining networks are similarly associated with higher order information processing. Now, another remarkable property is that they appear in fMRI data regardless of whether a stimulus is presented or not. So they're particularly useful for understanding brain activity during sleep. But if they're always present, how can they be useful for understanding changes in brain activity? The answer is that communication between them changes under different conditions. So we can use resting state networks as a kind of proxy for understanding what the brain is doing during sleep. For example, if daydreaming during wake is associated with a specific kind of communication between two resting state networks, and we see similar communication during REM, it's not unreasonable to infer that daydreaming might be taking place during REM. Or if we look at non-REM and we don't see these two resting state networks communicating, maybe dreams are not taking place in non-REM. So our paradigm was as follows. We recorded concurrent EEG fMRI from 33 subjects who slept in the scanner for 2 hours. We then used the EEG to sleep score the data and determine which MRI volumes were relevant to which stages. Now, we were fortunate to acquire a very rich data set with the greatest amount of REM fMRI data for healthy, non-sleep deprived subjects ever collected. So the key questions are as follows. How does communication between resting state networks change during sleep? And what can this tell us about sleep function and awareness? To give you a better sense of the analysis, let's simplify things by looking at only 3 networks. There are 3 unique lines of communication. We'll indicate them in red, black, and green. Now let's look at the green pair. Let's say that in wake, activity in each network is very similar to each other. This is reflected in a high correlation value. We'll call this high functional connectivity because we're making the assumption that distant regions that have similar activity are communicating with each other in order to carry out some function. We'll plot this as follows. State on the x-axis, in this case wake, functional connectivity on the y-axis. So in the case of wake, we have a high functional connectivity value represented with this green dot. Now let's say that in non-REM 2, the activity of this visual network on the right changes. So now we have a low correlation because their activity time courses are different. We assume that these regions are communicating less and likely not working together to carry out some function. We can then look at the other stages as follows. And finally, we can fit a line. This line represents the functional connectivity dynamics across wakefulness and sleep for that pair of networks. And we can do the same for the red pair and the black pair. So with these three lines, we have an idea of the functional connectivity dynamics of the entire system. Just to be clear though, instead of 3 networks, I looked at 14 networks with 91 unique connectivity pairs. But first, what might we expect? Well, we'll make a prediction based on EEG. We see a starting pattern in wake characterized by high frequency oscillations. We then see a progressive deviation away from this pattern towards slow waves during non-REM. And finally, we see a return to something that looks awfully similar to wake during REM. So this seems best fit by a curve like this, called a quadratic. So if we move to fMRI, we might expect functional connectivity to change like this. Importantly, we might also predict this, since we're simply predicting a progressive deviation away from some starting point, followed by a return to that starting point. So what did we find? Well, for about half the unique connectivity pairs, which are also called edges, there were no observed changes across the stages. These could be best described by a flat horizontal line. But for the majority of the remaining lines, they indeed match the predicted pattern. Here we see quadratics that curve down called convex on the left, and lines that curve up called concave on the right. Each line here represents one unique pair. And just to be clear, this box indicates connectivity in wake, and we then see a progressive deviation away from wake during non-REM 2 and slow wave sleep, with a return in the direction of the starting point during REM. So what does this tell us? Well, first it tells us that network functional connectivity dynamics largely match EEG dynamics. But it also tells us something else. Most of these lines curve downwards, as in the box on the left. What does that mean? Let's break this down. On the left, we see that connectivity starts positive and moves towards the opposite connectivity. Positive becomes less positive. On the right, it increases the same pattern. Positive becomes more positive. So what the left set of lines tells us is that most pairs of resting state networks are, at least initially, reducing their communication. If 2 networks communicate a lot during wake, then during non-REM, they talk less. This result is consistent with a homeostatic function for non-REM sleep. In other words, maybe the function of sleep is to homeostatically counterbalance wake-like communication patterns, so that in the following day, our brains are not overly biased towards some pattern of communication. This could help our brains better adapt to new situations the following day, because they're not overwired to a specific set of network relationships. Now, there's a final pattern in these results that's important here, though. Not only does the pattern of communication seem to go opposite towards 0, it actually crosses the 0. So positive doesn't just become less positive, it actually becomes more negative. The lines that do this are indicated in red, and these account for the majority of lines. So what does that mean? Well, it's further consistent with a homeostatic function, but it's also consistent with the idea that in slow wave sleep, connectivity isn't really best described as being reduced, which is how it is typically characterized. Instead, connectivity is best described as being different, flipped. So slow wave sleep might be better described as an altered rather than a reduced state of connectivity. Now let's take a closer look at the resting state network pairs involved here. As it turns out, the majority of these pairs, 13 out of 20 of them, involve higher order networks. Also, the majority of pairs involving the default mode network, 4 out of 6 of them, increase their connectivity during slow wave sleep. So why is this interesting? Well, sedation studies indicate that when you examine higher order networks, like the frontoparietal network shown here, during sedation, they actually reduce connectivity. On the left, you can see the frontoparietal network without sedation, and on the right, the same network in the sedated state. You can see that it's less intact, but sensory resting state networks, like the visual network here, maintain their connectivity during during sedation. And since sedation alters both conscious arousal and conscious awareness, this suggests that altering conscious arousal and awareness has a selective impact on higher order networks only. This further suggests that we can use higher order network activity as a proxy for conscious awareness and arousal. Finally, brain injury studies indicate that you can tell the difference between a vegetative state patient and a locked-in syndrome patient who has conscious awareness but no motor control solely by the presence or absence of the default mode network. On the left, we see that healthy controls have an intact DMN, and on the right, we see that locked-in syndrome patients also have an intact DMN. Vegetative state patients do not. This suggests that the DMN can be used as a proxy for conscious awareness. So together, these slow wave sleep results suggest that conscious awareness is increasing during slow wave sleep. So a state that is classically defined as a state of reduced consciousness may be better described as a state of altered consciousness. So what's the take-home message? Let's return to our original questions. First, what is the brain doing during sleep? If there's one answer I'd like you all to take from this presentation, it's this. During non-REM, brain network communication appears to change in a direction opposite to the network communication pattern found in wake. Second, why is the brain isolated from the environment during sleep? Well, in slow wave sleep in particular, we see altered network communication and indications of altered awareness. If, instead of being protected and isolated from the environment during such an altered state, we were to try to interact with the environment, we could be in trouble. And that concludes my talk. Thanks very much.
Hello, thank you to OHBM for this opportunity to present, given these unusual circumstances. My name is Nathan Cross, and I'm a postdoc in the Sleep Cognition and Neuroimaging Lab at Concordia University here in Montreal. And I would like to share some results from a study we collected investigating the impact of sleep deprivation on brain function and cognitive performance. So I'll jump right in with a quick statement that sleep is very important for overall brain functioning, as well as for our whole body. In fact, sleep is up there with the most important and vital factors we need to remain healthy, including the air we breathe and the food and water that we consume. Particularly for our brains, sleep has been shown to exhibit restorative properties that allow the brain to function optimally in the wake state. Some of these include maintaining synaptic homeostasis, flushing out of toxic waste through the lymphatic system, and memory consolidation. And it is well known that if we don't sleep, our cognitive performance suffers. And this is a well-cited study from 2003 that demonstrated the dose-dependent effect of sleep restriction on cognitive performances over time. Notably, vigilance and working memory. However, most cognitive domains have been shown to be affected by sleep deprivation. Each panel here displays 8 hours, 6 hours, 4 hours, and 0 hour sleep conditions over a 14-day study period. And what I would like to emphasize here is that after only 1 day of 0 hours of sleep, performance sharply drops to the point where after 3 days, this condition had to be stopped. However, few studies have investigated the state changes in whole brain activity with fMRI following sleep deprivation and their relationship to cognitive function. In a study from 2015, it was found that following sleep deprivation, there was an increased connectivity between the attention networks and the default mode network. Further, individuals who were resilient to declines in vigilance had greater levels of segregation between these networks at baseline. However, this was only after global signal regression was performed. When this regression was omitted, they reported a widespread increase in functional connectivity after sleep deprivation. Now, while this study demonstrated functional connectivity is clearly informative for performance changes, they did not further investigate this particular change after sleep deprivation and its relationship to behavior. And this premise is further supported by a more recent study finding a decrease in brain modulation following sleep deprivation. So we set out to investigate how an increase in brain connectivity following sleep deprivation might be related to cognitive performance. Our first aim was, how does the brain state change following 24 hours of total sleep deprivation? And is this altered following a nap? And our prediction here was that after sleep deprivation, global integration will increase and global integration will decrease following a recovery nap. Our second aim was, does the intra-individual change in brain connectivity inform on the individual level of change in performance? And our prediction here was that a greater increase in integration would result in a worse performance. Our study was a repeated measures crossover design. 20 healthy young individuals completed the entire protocol, which involved an habituation night in the lab, followed by either a normal night of 8 hours sleep or a night of total sleep deprivation. Following both nights, the subjects entered the MRI scanner, a 3T General Electric scanner, where they performed 3 cognitive tasks: the NBAC task of working memory, the attention network task, and the psychomotor vigilance task. In the sleep deprivation condition, the subjects were given a 60-minute opportunity to nap inside the scanner, and then the tasks were repeated. At all times, subjects were monitored with a 256 electrode high-density EEG system. And after 7 days, all subjects completed the other arm of the study. We implemented fMRI prep for the first level of our pre-processing, including basic features like registration and extraction of confound parameters. Then the data were further denoised through the XCP engine, which regressed out confounds such as movement in a conservative manner. And finally, we concatenated the 3 time series and regressed out the task blocks. This was performed to firstly lengthen the duration of the time series, providing more stable estimates and increasing our spatial resolution. And secondly, because this has been shown to provide a generalized functional connectivity estimate of the intrinsic brain activity similar to that of resting state data. We further sampled the bulb signal along the cortical surface, reconstructed by FreeSurfer on each subject's anatomical scan, and then into a standard space. Then the surface data was parcellated into 17 functional networks and further into 400 regional parcellations, which map to the networks on a one-to-one basis. And finally, we implemented a hierarchical model of functional integration to further probe the functional connectivity data. This model proposes that total integration can be divided into a measure of within systems plus between systems integration. So for example, at the network level, the total integration is given by the within network integration for each network, as well as a single measure of between network integration. And the total integration of a system can be directly computed from the covariance matrix, sigma, giving a quantitative measure of brain integration at each level. Moving on to the results, as expected, we saw a significant decrease in performance on all tasks following total sleep deprivation and a partial recovery following the nap. And this was observed for both the accuracy on the task as well as the reaction time. There was a widespread increase in functional connectivity following sleep deprivation, with 47% of edges increasing at a t-statistic greater than 2 after correcting for multiple comparisons. And the functional connectivity decreased following the nap to a lesser extent, with 16.7% of edges decreasing with a t-statistic greater than 2 after multiple comparisons correction. This widespread increase in connectivity coincided with an overall increase in total brain integration between baseline and the sleep-deprived condition. However, this did not decrease following the nap. The greatest increase in within-network integration was in the somatomotor cortex, likely driven by the fact that this was task-based imaging, and a similar trend was observed following the recovery nap, with the greatest decrease in integration occurring in the same regions. Most interestingly, however, was the relationship between the change in integration and the change in performance on the tasks. There was a robust, significant relationship between the increase in integration and decline in both accuracy and reaction time on both the attention and NBAC tasks. There was no relationship between integration and performance on the vigilance task, and this is likely related to the fact that we are only measuring cortical integration, and the nature of vigilance relates to brainstem and subcortical pathways. And these similar relationships were observed following the recovery nap, with those subjects who had a drop in total integration were better improved in their performance in the same tasks compared to the sleep-deprived state. So in summary, as expected, we observed a decrease in cognitive performances following sleep deprivation and an increase after the nap. Additionally, following sleep deprivation, there was a widespread increase in functional connectivity, corresponding to an increase in functional integration in the cortex. And this increase in integration was robustly associated with the decrease in performance on more complex cognitive tasks. Now, these findings suggest that prolonged wakefulness drives up levels of integration in brain networks, which impacts the individual's ability to maintain optimal performance during cognitive tasks. Now, it is uncertain what the physiological basis for this might be, and we are still investigating this. However, it appears related to the global signal fluctuation, which itself has been shown to be a consistent marker of arousal. And with that, I would like to thank everyone involved in this study, particularly those in my lab of Dr. Tan Deng Vu, and those in the lab of Dr. Christoph Grover, as well as those at the Perform Center who helped collect this data, as well as Dr. Habib Ben Ali, who helped us with the integration measures. Thank you.
Okay, welcome. I'm going to be talking about some of our efforts to decode depth of sedation from whole brain network structure, and more generally about some of our efforts to characterize changes in network structure across depths of unconsciousness. And to avoid philosophizing too much about what consciousness actually is, we're just going to define it operationally as a general state of unawareness, like non-REM sleep or anesthesia in this case. And neuroimaging research has often studied these states with the goal of describing features of brain activity underlying consciousness. And more recently, a lot of this work has sort of a very network flavor to it. And a common finding has been that during unconsciousness, whole brain networks exhibit a kind of fragmentation, or essentially the breaking up of brain networks into smaller, more isolated pieces. And our original interest was in seeing whether we could leverage some of these network changes to do prediction. But after going through all the work, we noticed something kind of interesting. And so I'm going to talk about the prediction briefly, but then I'm going to use it as a bit of a springboard to jump into something a bit more exploratory. And so we're reanalyzing data collected by Matt Husterton's group at Western University. And we have 12 5-minute resting state scans, so 2 at each of 6 doses of isoflurane. And we're going to be looking at whole brain cortical networks. So we parcellate the cortex into 174 ROIs using the Van Essen Atlas. And for each scan, we get a correlation matrix containing the pairwise bold correlations between ROIs. And we're going to look at both the correlation matrices themselves and at graphs made by thresholding those correlation matrices. And so just to get a sense of what some of those networks look like, and just to replicate some effects that have been seen in the literature, we're going to make some graphs just by thresholding the correlation matrices by just using a fixed magnitude threshold. So in this case, just by significance. And we can see that the networks become sparser, the larger number of communities, they become more modular, more small world as dose increases. And so for those who may be less familiar with some of these measures, the main takeaway is that these are all things that are commonly interpreted as reflecting a kind of fragmentation of these networks. And our thought was that given how clear and consistent these network changes are, it should be possible to decode depth of sedation from resting state functional connectivity. So I'm going to summarize our general approach here, but I'm not going to go into too much detail, mostly because in the process of doing these predictions, we noticed something that kind of led us down another rabbit hole, which I'm going to talk more about. So I'll just summarize our basic pipeline briefly, and then I'll show the results. And so our goal was to learn to predict dose of isoflurane based on a whole brain resting state correlation matrix. And the general machinery was modeled after the approach used by Barenshon et al. And it's basically a support vector machine that uses a kernel based on the Riemannian metric on the space of covariance matrices, which just intuitively is a natural way of describing similarity between covariance matrices. And prior to doing this, we center each subject to remove static subject differences, since we know, say from the Midnight Scan Club, that quite a lot of recent work, that functional connectivity is dominated by these kinds of stable subject-specific effects. And we just wanted to remove those effects by centering them out. And this general approach seems to work quite well. We can assess the model by leave one subject out cross-validation. And the predictions, if you look at the top figure, are generally quite accurate. This is the dose against the predicted values. So perfect prediction would lie along that diagonal line. And it's good, but we do consistently underpredict at the highest dose. So we asymptoted around 2% isoflurane. But if you look at the bottom plot, which shows the root mean squared prediction error for each subject, we do better than just, say, a reference model, which only predicts the mean dose, which is that dashed gray line. So we can do prediction, and we can do it with a relatively simple model, so that's fine. But when we sort of started poking at this to figure out exactly what was driving these predictions, we got sidetracked by something that's kind of interesting, which is that we don't actually need the whole brain. When we started retraining the model just on randomly selected sets of ROIs, we found that we could get comparable performance with as few as 25 randomly chosen regions. So the plot at the right shows the root mean squared error for a few hundred models trained on random subsets of the whole brain atlas. And we start to see asymptote right around 25 regions. So whatever it is that's being decoded can't be something localized. It has to be decodable pretty much everywhere. And so we started thinking not just about the prediction, but about the network effects that we saw earlier. And one interesting observation that we made, if you look at that top figure, is that increasing dose seems to be associated with an overall decrease in correlation magnitude. And in fact, that turns out that this isn't localized either. It's present pretty much everywhere. So it's present in primary sensory regions, like the auditory, somatomotor, and visual regions, and it's present in higher order association areas. And the reason we care about this is that, remember, we often work with networks constructed by thresholding correlation matrices. And so overall changes in correlation strength can still result in changes in graph properties, even when the actual pattern of correlations is the same. And so this raises an interesting question, which is, do these network changes, or this fragmentation that's often described in the literature, reflect an actual change in network structure, or is it just an artifact of this kind of global reduction in functional connectivity? And so to probe this a bit further, we created some surrogate data sets by holding either the pattern of correlations or the magnitude of the correlations constant. And so in what we'll call the constant structure data set, we replaced each correlation matrix with a copy of the average correlation matrix at the subject's lowest dose, and just rescaled it to have matching average correlation magnitude as the real correlation matrix. And in the constant magnitude data set, we rescale each correlation matrix to have the same average magnitude at each dose, so that only the structure was allowed to vary. And then we thresholded them and looked at the same network statistics as before. And when we do this, we see that our pattern of effects are completely consistent with a constant network structure and just an overall decrease in correlation magnitude. These are the red and black lines. So the black are the effects on the real data set, and the red is the effect of the data set with the correlation structure held constant and only the magnitude allowed to vary. And these effects are completely abolished if you hold the magnitude constant. And so this kind of fragmentation, it seems, can occur even when there's no actual change in network structure. And just to measure this more directly, if the general structure of the network is preserved across dose, then the network structure should be just as similar to a scan at a different dose as it is to another scan at the same dose. And so we split the data in half, and we compared the correlation structure at each dose with either the other scan at the same dose or with the lowest dose. And we do this by comparing the principal components. So specifically, we take the top 5 components, since these are the components whose eigenvalues seem to be most strongly affected by dose. And we look at the distance between the subspaces spanned by those components, called the Grassmann distance in that bottom figure, which is essentially just a way to measure how much the principal components differ. And it turns out that it doesn't really matter whether you compare one scan to another at a matched dose or to another one at the lowest dose. The structure seems to be just as similar or just as different. And so the correlation structure seems to be more or less preserved across dose. And so an obvious next question is, what does this structure look like? And so to visualize it, we use common principal component analysis to jointly decompose the full set of correlation matrices. And just to take a quick look at some of the leading components, if you look at component 1 there on the left, we seem to have a gradient separating posterior visual areas from the rest of the cortex. 2 seems to be a gradient separating somatomotor regions from the rest of the cortex. 3 and 5 seem to be these mirror symmetric components separating frontal parietal regions in one hemisphere from the rest of the cortex. And most importantly, all these components seem to be maximally expressed at the lowest dose. And they just decrease almost monotonically as dose increases. And they asymptote right at around 2% isoflurane, which is incidentally right about where our predictions top out. And so just to summarize, our general impression at this point is that isoflurane, at least, is associated with a global reduction in whole brain functional connectivity. And this fact alone is sufficient to explain these kinds of fragmentation results that are often observed in thresholded correlation networks. And in contrast, we find that a lot of network structure is actually preserved across depth of sedation. It just becomes weaker or muted or noisier. And so graph metrics alone can be a bit misleading, and there really is a need to examine network structure in fine detail if we want to understand what's really going on. And there's some challenge to disentangling real structural changes or local changes from these kinds of global effects. And we will note, just to keep in mind, that isoflurane itself is a vasodilator, while many other anesthetic agents are not. And so it's important to distinguish between effects that may be unique to an individual anesthetic, say a vasodilator's effect on hemodynamic responses, which may be more global, versus the common effects across many different kinds of compounds, which actually account for their anesthetic effects. And in fact, there's even some recent work out of George Mashour's group in Michigan suggesting that consciousness may even be dissociable from cortical connectivity. And so there are some interesting questions about whether these kinds of functional connectivity networks are actually sufficient to completely describe the network correlates of consciousness, or whether we might need sort of more fine-grained analyses or more principled models. So I'll just end on that kind of exploratory note and those kinds of open questions that might be important for work in the future. Thank you.